And without membranes, there are ribosomes and centrosomes. And with membranes, we have again two types. With single membrane, single layered membrane and double layered membrane. So today we will stick with cell organelles without membranes, that is ribosomes and centrosomes. Ribosomes are granular structure, first observed under electron microscope, composed of RNA and proteins without membranes. The first person to observe the ribosomes was Claude, and it was named by Palade as ribosomes. Eukaryotic ribosomes are of two types. Ribosomes are ATS, which are again of two types. It is divided into two subunits. In fact, not of two types, but made up of two subunits, which are 40S and 60S. Eukaryotic ribosomes are ATS, and they are made up of two subunits, 40S and 60S. Prokaryotic ribosomes are of 70S type, which is again made up of two subunits, 50S and 30S. So they are also made up of two subunits, that is 50S and 30S. Now, what does S stand for? A little explanation is required here. S stands for Sedberg's coefficient, or it is also known as sedimentation coefficient. Sedberg's coefficient. S stands for Sedberg's coefficient, which is also known as sedimentation coefficient. It is also known as sedimentation coefficient, which is an indirect measure. It indirectly measures the density and size of the cytoplasmic organelles. It measures indirectly the density and size of the cytoplasmic organelles. So during centrifugation, when we actually separate the cytoplasmic organelles, what we normally do is centrifuge गरिन्छ। जस्तो सेन्ट्रिफ्युज भनेको कस्तो खालको भन्दाखेरि हामीले दुध पेल्ने मेसिन देखेको छ, झन्डै झन्डै त्यस्तै खालको नै हुन्छ, तर त्यसमा चाहिँ डिफ्रेन्ट टेस्ट ट्युब्सहरु झुन्डाउने जग्गा हुन्छ। अनि ड्युरिङ सेन्ट्रिफिकेसन डिफ्रेन्ट स्पिड्समा डिफ्रेन्ट डेन्सिटीको अर्गानेल्सहरु छुट्टिँदै जान्छ, हो त्यसलाई चाहिँ भन्छ सिडबर्ग्स कोएफिसिएन्ट अर सेडिमेन्टेसन कोएफिसिएन्ट। सो इन्डाइरेक्टली त्यो डेन्सिटी अनुसार, त्यो साइज अनुसार त्यो साइटोप्लाज्मिक अर्गानेल्सहरु छुट्टिँदै जान्छ, त्यसलाई चाहिँ भन्छ सेडिमेन्टेसन कोएफिसिएन्ट अर सिडबर्ग्स कोएफिसिएन्ट। It is somewhat like a centrifuge. It is a centrifuge. A device is the centrifuge, and the process is called centrifugation. Now, ribosomes are a single unit. During protein synthesis, in some of the cases, number of ribosomes join together. Then it is called polyribosomes. Number of ribosomes join together like this. They work as a single unit, and they are basically related with polycistronic mRNA, which helps in the process of making number of proteins that is required as enzymes in different biochemical reactions. यसलाई चाहिँ भन्छ polycistronic mRNA हुन्छ. त्यसमा चाहिँ थुप्रै कोड्सहरु एकैचोटी हुन्छ that is required for a single chemical reaction. त्यस्तो खालके जब थुप्रै राइबोसोम्स यसरी एक लाइनमा जोडिएर mRNA त्यससँग जोडिन्छ त्यसलाई चाहिँ भन्छ polyribosomes. यो जुन चाहिँ भाग छ यो चाहिँ हो mRNA. अनि these are the ribosomes which work together and as a unit. त्यसलाई चाहिँ भन्छ polyribosomes. Number of ribosomes stick together. Now the function of the ribosomes. We are very much aware as far as the function of the ribosomes is concerned because we have talked about ribosomes, the basic function of ribosomes in the junior classes as well. The basic function of ribosomes, it helps in the process of protein synthesis and metabolism of fat. It helps in the process of protein synthesis. यहाँनिर पनि हामीले भन्दैछौँ mRNA जब जोइन हुन्छ mRNA कहाँबाट आउँछ it comes from basically a copy of DNA. DNA को जुन चाहिँ इन्फर्मेसन छ DNA त जहिले पनि न्यूक्लियस भित्रै बस्छ त्यसबाट बाहिर जुन चाहिँ इन्फर्मेसन आउँछ त्यो चाहिँ mRNA ले लिएर आउँछ अनि त्यो mRNA मा भएको इन्फर्मेसनलाई ट्रान्सलेट गरेर प्रोटिन्स बनाउने काम जुन चाहिँ हुन्छ त्यो चाहिँ राइबोसोम्सले गराउँछ. That is why ribosomes helps in the process of protein synthesis and it also helps in the process of metabolism of fat. यो जुन चाहिँ इन्फर्मेसन आउँछ भित्र DNA बाट आउँछ अनि त्यो चाहिँ केले लिएर आउँछ भन्दाखेरि mRNA अनि mRNA आएर चाहिँ यसरी चाहिँ राइबोसोम्ससँग जोडिन्छ अनि यो राइबोसोम्समा जुन चाहिँ के रे mRNA मा जुन चाहिँ इन्फर्मेसन छ त्यसको आधारमा यहाँनिर चाहिँ प्रोटिन्स बनिँदै जान्छ. त्यसको आधारमा चाहिँ प्रोटिन्स बनिन्छ. यसको एक्जाम्पल हामीले polyribosomes को एक्जाम्पल चाहिँ हामीले the enzymes that is required for the metabolism of glucose metabolism जुन चाहिँ छ त्यो ग्लुकोज मेटाबोलिजममा जुन चाहिँ एन्जाइम्सहरु चाहिन्छ ग्लुकोज ब्रेकडाउन भएर हामीलाई 38 मोल्स अफ ATP हरु जुन चाहिँ बनिन्छ हो त्यो प्रोसेसमा चाहिँ यसरी चाहिँ नम्बर अफ एन्जाइम्स आर रिक्वायर्ड अनि त्यो चाहिँ polyribosomes द्वारा बनिन्छ. So basically here we need to remember the main function of the ribosomes is it helps in the process of protein synthesis and it also helps in the process of fat metabolism. Now we move on to the next single membrane bound cytoplasmic organelle, that is centrosome. Centrosome with centrioles. Centrosomes with centrioles. This centrosome is made up of two words. Centron meaning center and zome meaning soma, which we already know means body. So it is basically centrosome is made up of two words, centron and zome. Centron comes from the words meaning center and zome means soma and soma means body. This centrosome has two cylindrical structures inside it which are basically known as the centrioles. Two cylindrical structures. Duita cylindrical structure which are placed at 90 degrees to each other. Duita cylindrical structure. This is known as the centrioles. Duita cylindrical structure inside the centrosome, which is known as centrioles, and it is surrounded by a clear cytoplasm, which is known as centrosphere. So centrosome basically is made up of two cylindrical centrioles at the center and centrosphere, a clear cytoplasmic region around the centrioles, which is known as the centrosphere. Alright. Now, These centrioles are basically made up of 9 cylindrical peripheral fibrils. These centrioles look like a cartwheel. They look like a cartwheel. Cartwheel भनेपछि जुन चाहिँ गोरु गाडीको जुन चाहिँ चक्का हुन्छ त्यस्तै खालको नै देखिन्छ। अनि यसमा चाहिँ जम्मा 9 वटा चाहिँ evenly spaced 9 वटा evenly spaced कस्तो खालको भन्दाखेरि 1 2 3 4 5 6 7 8 अनि 9 यसरी 9 वटा ट्युब्युल्सहरु चाहिँ प्रेजेन्ट हुन्छ which are evenly spaced अनि एक अर्कादेखि एकदमै इक्वल डिस्टेन्समा हुन्छ। evenly spaced peripheral tubules, यसरी peripheral means at the boundary, towards the boundary, and they are made up of tubular proteins. These peripheral tubules, fibrils, are made up of tubular proteins. Each peripheral fibers are made up of 3 micro subtubules, 3 subtubules, triplets of micro subtubules. Like, let me explain it through the diagram. First, we have the central rod, जुन चाहिँ हामीले त्यो गोरु गाडीहरुको चक्कामा पनि देख्छौं यसरी central rod छ। त्यहाँदेखि बाहिरको चक्का छ, यसरी चक्का छ यसरी। नाउ यो चक्कामा इक्वली स्पेस्ड 2 5 6 7 8 यसरी 9 वटा स्पोक्सहरु जस्तो प्रेजेन्ट हुन्छ। 9 वटा यसरी स्पोक्सहरु जस्तो प्रेजेन्ट हुन्छ। अनि 2 3 4 5 6 7 8 9 evenly spaced, यो दुईटाको बीचमा evenly spaced 9 वटा यो पेरिफेरल ट्युब्युल अब बनाउन गइरहेको छु, यसरी छ। पेरिफेरल ट्युब्युल्स यसरी छ 9 वटा। peripheral tubules, and these 9 peripheral tubules are further subdivided into subtubules. The peripheral fiber is subdivided into number of tubules like this, or we can say into 3 tubules. That's why it is called triplets. यसरी चाहिँ जोडेको हुन्छ। यसरी जोडेको हुन्छ। What we have the center is the central rod. Then these are the peripheral tubules. Which are further made up of 3 subtubules. These are the peripheral tubules which are further made up of subtubules. These are the subtubules. And these are the spokes. So here what we see is this centriole looks almost like a cartwheel, right? Here the हामीले अब sectional view we are studying the sectional view, but this is a totally a cylindrical structure like this. अनि यो सबै tubules चाहिँ यसरी equally spaced यसरी हुन्छ. सबै नै equally spaced यसरी tubules हुन्छ. यो सबै equally spaced tubules यसरी equally spaced tubules हुन्छ. Now we talk about the function. This is the structure of the centrioles. Here what we need to remember is the one at the center is called the central rod. Then we have the spokes. Then we have 9 peripheral fibrils. These peripheral fibrils are made up of proteins and they are further subdivided into subtubules. Each are made up of 3 subtubules. That is why they are called triplets. Triplets of microtubules arranged longitudinally. Now the function of the centrosome. The function of the centrosome, basically what we have studied in the junior classes is that it helps in the process of formation of spindle fibers from the opposite poles. यो त हामीले कुरा गरिसकेका छौं. जस्तो सेल डिभाइड हुन्छ, दुईटा पोलमा सेन्ट्रोसोम जान्छ, त्यहाँबाट चाहिँ यसरी चाहिँ spindle fibers आउँछ. So it helps in the process of cell division by formation of the spindle fibers. That is the first important function. The other important function of the centrosome, it helps in the formation of basal body from which arise the cilia and flagella. Cilia just the paramecium मसिनो cytoplasmic outgrowths हुन्छ which helps in locomotion. त्यसरी नै flagella euglena मा हुन्छ जुन चाहिँ locomotion मै help गर्छ. हो तिनीहरु बनिनुको लागि चाहिँ यो basal body चाहिन्छ. त्यो basal body बाट निस्किन्छ अनि that basal body is made up of centrioles or we can say centrosome. Spermatozoa, like in the case of human beings, the spermatozoa has to swim through the vaginal fluid to reach the eggs. त्यो जुन चाहिँ स्विम गर्नको लागि त्यसको टेलमा जुन चाहिँ मोसन चाहिन्छ त्यो टेल बनिनुको लागि पनि के अरे the tail of the spermatozoa is also formed with the help of the centrosome. Another most important function of the centrosome is it helps in the process of the movement of the sperm from the vaginal fluid to reach the eggs. Now, this is about the 2 cytoplasmic organelles with and without the outer membrane. That is ribosomes and centrosomes. Now we go on with membrane, cytoplasmic organelles with membranes. And the first that we are going to discuss here is with single layered membrane. It has only one layer of membrane around the cytoplasmic organelle, single layered membrane. And in that we are going to talk about, first that we are going to talk about is the Golgi body. This Golgi body was first discovered by a scientist named Camillo Golgi. That is why the name Golgi body. It was giving respect to the person who discovered. The structure has been named after his name. That is why the name that we study now is Golgi body, derived from the name of the discoverer. And this was discovered by Camillo Golgi in 1898. That is pretty long time. 1898 मा चाहिँ यो discover भएको हो by Camillo Golgi. This Golgi body is placed in an eukaryotic cell, this is the nucleus. In a eukaryotic cell, the Golgi body is placed near the nucleus, and it is made up of stacks of sac-like structure, which is called the cisternae. This is cisternae, and it is convex in shape. These are flat, dish-shaped structures which are stacked one on top of the other, and these structures are called the cisternae. This is the most prominent structure of Golgi body. That is, it is made up of flattened sac-like structures or dish-shaped structures, almost convex in shape, and it is called the cisternae, which are parallel to each other. They do not join each other. They are parallel to each other, and associated with this, they are very close to the nucleus, and they are associated with this are small packets called vesicles. Small packets called vesicles. These vesicles are basically the structure that pinches out from the Golgi body after the concentration of the substance that needs to be secreted. In plants, these structures, the Golgi body in the case of plants is called dictyosomes. In the case of plants, it is known as dictyosomes. So Golgi body is basically made up of cisternae and vesicles. Cisternae and vesicles. Let's enlarge this structure of the Golgi body like this. It is convex in shape. Number of layers of decreasing size. They run parallel to each other. These are called the, as we have already talked about, these are called the cisternae, and this side, this face, the outer convex side, is called the forming face, and the inner side, the concave side, is called the this forming phase is also known as cis phase, and the inner side of the cisternae, the inner layer, is called the maturing phase, or it is also called trans phase. The convex phase is called the forming phase or the cis phase, and the inner concave phase is called the maturing phase or the trans. Now from the maturing phase, number of small packets arise. These are known as vesicles. These are known as the vesicles. The function of the Golgi body is basically packaging. Whatever substance needs to be sent out of the cell is first taken to the Golgi body, where it is concentrated and packed in small packets called vesicles, and these vesicles helps in the transport of this substance to the place where it is required, or transport of this substance outside the cell. So basically we can say Golgi body is a packaging unit. It packs up whatever needs to be sent out, and all the substance that needs to be sent out is concentrated in the cisternae, after which these small packets, the vesicles, which are also known as the secretory vesicles, are formed. These secretory vesicles, when they are large, then they are called secretory vacuoles. They are known as secretory vacuoles. Now here we need to understand one more important thing, that is GERL system. GERL system. This is basically Golgi body, endoplasmic reticulum, and lysosome combined together as a unit is called the GERL system. They work as a unit. And they work together in the process of secretion. All these structures, the Golgi body, endoplasmic reticulum, and the lysosome, they work together as a unit in the process of secretion. Secretory vesicles contain acid phosphate and enzymes. The secretory vesicles contain acid phosphate and enzymes, and this is concentrated in the cisternae and it is sent out to the place where it is required. GERL system is a part of smooth endoplasmic reticulum involved in the formation of lysosomes. GERL system is normally associated with the formation of lysosomes.